Good evening, everybody, and thanks for coming tonight to our last session of the Chronic Waste and Disease webinar brought to you by the University of Minnesota Center for Prion Research and Outreach. I'm Mark Schwabenlander, and I'm the CWD Research and Outreach Manager here at MinPro. I'll be moderating tonight's webinar session like the last three. A few quick Zoom webinar housekeeping items. Get my PowerPoint moving here. There we go. The session will be recorded and will be available on the MinPro website afterwards tomorrow. The attendees' cameras and microphones are off during the session. If you have any questions, please enter them in the Q&A tab at the bottom of the screen anytime during the presentation, and we'll go through those questions after the presentations are done. If you have not interacted in Zoom, there are steps for the Q&A tab. If you follow along the green circle here, the Q&A tab is at the bottom of your Zoom screen. If you don't see it, if you hover your mouse over the bottom of the screen, it will appear. If you click on that, it'll bring up that screen, and next you can type your question into the box. If you want to send that question anonymously, you have that option. Just click the little tab next to the Send Anonymously. And then finally, once you have that question in, you can send it our way. If you're listening to this session via telephone or watching the recording, you can send questions to our MinPro email and we'll happily answer those. So without further ado, our last session will be presented by Eric Hildebrand. Eric is a wildlife health specialist for the Minnesota Department of Natural Resources, and he'll be discussing Minnesota's CWD management in wild deer. Plus, we have a bonus discussion from Dr. Chris Janelle, a Minnesota DNR research scientist, on the DNR Southeast Minnesota Deer Movement Study. Thanks, Eric and Chris. I will unshare my screen and you can take it away.
All right. All right, Mark, let me know when you can see the PowerPoint.
We are good.
All right. Well, good evening, everyone. And we'll jump right into things. So chronic waste and disease, CWD, just a quick overview of what it is. It's a slowly progressive brain disease of cervids, so deer, elk, moose, reindeer. It belongs to a family of diseases known as transmissible spongiform encephalopathies, or TSEs, or also it's known as a prion disease. This is not caused by a virus or a fungus or a bacteria, but rather a misshapen protein. And the way that this disease can be transmitted among deer is mostly through saliva, feces, and urine. And I do want to point out the two pictures that you can see here. Both these animals are confirmed CWD positive. And notice the top animal is in a preclinical stage, whereas the bottom photo shows a clinical symptomatic animal. And just a little outline for tonight's topics. We will talk about new areas that we have with increased risk for CWD in Minnesota. Two of them are due to positive deer farms, and another one is due to a positive wild deer that was detected. We'll talk about our upcoming surveillance for 2020, what we're seeing in our persisting infection area of Fillmore County, and then we'll move into Chris Janelle's presentation with the Southeast Deer Movement Study, and then we'll take any questions towards the end. And just some fun stats on the side. Tested a lot of deer last year with 36 additional new positive wild deer. Since 2002, we've tested more than 90,000 deer in Minnesota. Currently, we're sitting at 91 positive deer in 6 different counties. And here's a map that kind of shows the locations of where the 11 positive captive cervid facilities have been detected since 2002. So those green counties are highlighted. And then the red dots are where we have our 91 positive wild deer to date. We do have persisting disease versus initial detection. So I'll get into that in a little bit. So new this year, we have our 2 positive deer farms that were recently detected this year, and that was West Central Surveillance Area, or Douglas County area, And then our East Central surveillance area, or Pine County area. And we have a positive wild deer that was detected in March, and that was in Dakota County, which we're now a new deer permit area was created, DPA 605. That is designated our South Metro CWD surveillance area. And then we have our ongoing efforts in the Southeast, which is where we have our persisting infection, and our North Central management zone, deer permit area 604, which is still under our initial detection. We found one positive deer. It was since then we've not detected another positive after testing an additional 9,000 animals in that permit area. I do want to note, last year was our third and final year at the time for our central surveillance area. We had 3 consecutive years in Meeker County, Minnesota, where a positive deer farm was detected, and we conducted 3 consecutive years, all not detected results, and so the surveillance area has been dissolved for this upcoming fall. So the 3 areas that are new this year, we do have some goals that are established for these areas. In these surveillance areas, East Central, we have a goal of 1,000 samples, and self-service sampling stations will be made available starting with firearms season, November 7th. Those stations will be available for hunters until the goal is reached. In the West Central surveillance area, it's a different approach that we are piloting this year, and other states are utilizing this risk-based sampling model. And this model designates each cohort of deer and assigned a certain amount of points. So such as an adult male is worth more points than a fawn male would be. And so we have certain point goals established per DPA based on certain risk factors. These stations are going to be available, or I should say have been available since October 15th now. And once the point goals are reached in both deer permit areas, those stations will be removed. And then we get over to the South Metro surveillance area. And so in the gray area that you can see, that has a goal of 750 deer. And those stations, just like the East Central, will be made available starting November 7th until the goal is reached. And I do want to point out that in these areas, all the gray areas here, there are no carcass movement restrictions. And I'll get into more of that here coming up. The Southeast and North Central management and control zones, those boundaries are the same as last year. Again, we have persisting infection that we're seeing in the Southeast management zone. We have all stations available during all seasons, archery, firearms, and muzzleloader. And here we do have carcass movement restrictions in place. We do have dumpsters and quartering stations that are made available to hunters within these zones. And so carcass movement restrictions, these are the 3 areas in the state. The new permit area 605 is a CWD management zone. So all 3 of these areas, we have detected disease in wild deer. And so we know that the disease is in wild deer here, so we want to reduce any potential for this disease to spread through hunter movements of deer carcasses. And that's why we put these carcass movement restrictions in place. Because the prions reside in the neurologic tissue, so such as the spinal column or brain tissue in the infected animal, by properly disposing of those high-risk parts, we can help mitigate the spreading of the disease and further transmission of this disease. So a dumpster program was initiated. Last year was the first year. We continued it this year. So last year, over 200 tons of deer waste was generated in these areas, and it was very costly, just at about $200,000. And the dumpster locations are located on the DNR's website. So I urge viewers to take a peek at where these locations are, and some approved methods for disposing of positive CWD deer We do have alkaline digestion, which is available at the veterinary diagnostic lab at the University of Minnesota. Line landfills, and also incineration if the temperatures do exceed 1500 degrees. So expanded deer and feeding attractant use bans. I mentioned that the 2 new areas this year where there were positive deer farms detected. So East Central and West Central, or Pine and Douglas County areas. You'll notice that's where we have no deer feeding. And then where we have detected disease in wild deer, you'll see we have no deer feeding or attractant use. And so we know that the disease can spread through transmission between deer, and by reducing any artificial concentration of deer that humans can create, this will put a break in that system where this disease can be transmitted further. And so even though attractants are banned, attractants such as Doan heat or a mock scrape drip, where it has cervid gland oil and whatnot, or blood, we know that certainly, yeah, CWD prions can be in deer urine, but that's not the motive behind the attractant ban. We don't want, for example, the mock scrape drip to bring in multiple bucks to one site, where if one buck is positive for CWD, the likelihood that those additional bucks that come to that one spot where that mock scrape drip is, it increases the likelihood that those other animals can become positive. So some changes for this upcoming fall to our surveillance efforts. We have shifted from in our CWD management and control zones. Last year, we were under mandatory sampling that hunters had to abide by if they harvested a deer in these areas. This year, it is voluntary for hunters, and this is to minimize any social interaction with COVID-19 concerns in mind. Previously in last year, we had in-person check stations where DNR staff and university students would interact with hunting parties and hunters. And as you can see in the photo to the right, a lot of congregation at the bed of a pickup where we would get data, collect the sample. So it was decided to shift to voluntary and not put hunters at risk if they don't feel they would want to be at risk. So self-service sampling stations are utilized this year and not the in-person check stations. However, carcass movement restrictions for all deer, including fawns, are mandatory and will remain in effect for the disease management and control zones. And then once the deer heads are picked up at these self-service sampling stations, they go back to a central extraction site where DNR staff and university students will collect the data and send it up to a digital database, and then pull out the lymph node samples to send off to lab for testing. And so self-service sampling stations, here on the right, you can see an example of one. We have everything needed that a hunter would need to cut the head off, where there's a saw available. There's a map that you can't quite see on this side, but on that 4x4 sheet of plywood, there's a laminated map that helps the hunter orient where they harvested the deer to provide township range and section level data. There are carcass tags inside the box that is very straightforward for the hunter to provide anything from hunter's name, a phone number, the MNDNR number, and that allows the DNR, if it is positive, we have a phone number, we can contact the hunter to let them know that their animal was positive. And also, if they provide their DNR number, that allows the hunter to go to the DNR website and check their own test result. So deer heads are picked up 3 times a week during archery season, and then during the firearm season, we increase that because harvest is much more during firearm season. But it's hard to predict what hunter behavior would be. So we geared up based on a range, 30 to 70% compliance by hunters. So diving into Southeast Minnesota, and this again is where we have persisting infection, and you'll see that from the initial area, we are starting to see some spread and out to new areas within the management zone. Those being Winona County and also Houston County. In the Winona County area, immediately surrounding a positive deer farm, we now have 9 cases in the wild deer in that area. Then in Houston County, you can see there's it looks like just 1 or 2 blue dots that you can see on the map, but there are 4 infected deer that were collected off the same section in the Money Creek area. And so when we look at what we call our core area, this is where the disease seems to be at a higher prevalence, and 50 of the 89 deer that are positive in southeast Minnesota have come from this 8x8 core area near Preston, Lanesboro. This area has been the focus of USDA Wildlife Services agency culling operations. And recently, the past winter 2020, agency culling was conducted here along with in the Winona County infection that I pointed out previously. Seems to appear that hunting pressure has been consistent between all years since the disease has been detected in fall 2016 in this core area. And so the agency culling operation, this is one of the most effective tools that we can use to really combat this disease. The focus for this effort has been on the first priority sections where we have confirmed positive deer. It's not just the entire deer permit area, but rather the known infection section where that deer came from. And then we reached out to landowners in the first and second priority. Anyone that owned 10 acres or more, we sent them a letter and explained the project, explained why, and offered the phone numbers and emails if they wanted to participate, or if they had any questions, they could reach out directly to us and get a hold of us. So this past winter, there were 463 deer removed, and of those, we had 7 additional positives. There was 1 additional positive that was found dead by one of the USDA staff members, but that was not harvested by USDA. It was just found dead. And so, with the winter 2020 continued, all the deer that were removed by USDA Wildlife Services went to a local meat processor, and after they were sampled for CWD. That meat processor broke the deer down and processed it into steaks, roasts, and then trim, nothing such as sausage or brats or anything like that. But then when those animals came back as not detected from the lab, then those not detected venison went to what we call the Share the Harvest program. And this program, any public member can get signed up on the DNR website to receive venison. So approximately 360 of these deer went to the Share the Harvest program, and just about over 100 deer went back to the participating landowners that allowed removal of deer from their property. And so when looking at CWD prevalence, I mentioned we have persistent infection in the southeast. Here's a graph that shows where we're at. And currently, in the boundary is Deer Permit Area 603, which is no longer in existence right now, but for this purpose, that's the boundary that's set. We're still at 1%, a very, very low percentage of infection in Minnesota. And I want to make this slide very prominent to viewers, because when you look at other states, such as Wisconsin or out west, there are states that have infection upwards of 40%. And again, Minnesota, we are still sitting at a very, very low percent at 1%. By using tools to aggressively manage this disease, I mentioned earlier the carcass movement restrictions, the feeding and attractant bans, using tools such as agency culling that is very, very directed at where those known positive locations are. That's a very good way to keep this disease at a very low prevalence in our deer herd. We conducted aerial deer surveys. Here is the boundary of deer permit area 603, which again is no longer in existence now. It's a kind of a combination of deer permit area 647 and 648 right now. But you can see the change in the core area. You know, deer densities are still very, very high, and so you know, even with agency culling and offering liberalized tags, unlimited disease tags to hunters, these deer densities are still at high levels. And so what does this mean for Fillmore County, where we have persisting infection? It looks like we will be living with CWD in this immediate area, as it is persisting at a low prevalence within the local deer herd. We are always encouraging hunters to get their deer tested. There are CDC guidelines that currently do not recommend any person to consume a known CWD positive animal. We also encourage landowners and hunters to help combat this disease, properly disposing of carcasses and reduce any sort of artificial concentrations of deer. These are the ways that this disease can spread. And by removing positive deer off of key pieces of property, we do see there are sanctuaries, quote unquote, of high densities of deer in local areas, which, you know, these sanctuaries can then further spread this disease within that deer herd. By implementing strategies and actions laid out in our CW management plan, I encourage folks to check it out. It is posted on our DNR CW website in PDF form, so take some time and read the plan. Some great information in there, and if you have questions on what's to come, it's laid out nice. So take a look at the plan. Public engagement and outreach. The public needs to know what's going on, and so the only way to do that is by getting communications and outreach out to those public figures, hunters, landowners, wildlife watchers. So the test results from this upcoming fall, that will show where agency culling should focus. Again, we only focus those efforts on where known positive animals are located, and then within the first and second priority sections around that, that known positive. So we also need to keep the prevalence low in these areas where we have persistent infection. By doing this, hunters need to keep hunting, as they are one of the most key roles to keeping deer populations from increasing too high. And so with that, I would like to shift over to Chris with the Southeast Deer Study.
Hey, thanks, Eric. Great job, Eric. Awesome overview of our surveillance and management efforts for CWD Minnesota. Thanks to Peter and Mark for having us. It's a pleasure to be here and give these talks to folks. Eric did a great job of introducing CWD and what we've been doing with CWD management in Minnesota. This is a more detailed look at a Southeast deer movement study that we started, and it started. We started thinking about this actually right after we detected CWD in the fall of 2016 in Fillmore County. And the basic objectives of this study were to document dispersal patterns and look at activity ranges of particularly juvenile males and females, so fawns, and in some cases adult males within southeast part of Minnesota, surrounding the then 603 permit area, which enclosed all of our known detections at the time. And we wanted to utilize this information to really understand and map and inform corridors of possible CWD spread. We knew early on that we wanted to be forward thinking and try and head this off as much as we could and learn about possible movements of deer and how those are related to movement of CWD in the landscape, and use that information to inform our management and surveillance. Another thing we wanted to look at was determining cause-specific mortality from deer that we actually put GPS collars to track. And we could use that information as a secondary objective to inform our population modeling. I won't be going over that this evening. I'll just be talking about our movement pieces. But next slide, please. But we had a grant opportunity by the Legislative Citizens Commission on Minnesota Resources, the LCCMR, and we got about $450,000 from their Natural Resources Trust Fund to basically be the seed money to get this project off the ground. We've also actually put quite a bit of further funding in by the Minnesota DNR itself, and we have a website devoted to this project that you guys are welcome to check out. It's easy to find, and here's the website address here on our page here. Next slide. So the study area is basically southeastern Minnesota, and it covers an area of approximately 3,000 square miles. And as you can see by that black outlined area on the map there, we split up the study area into different quadrants so that we could try and equally apportion deer that we capture by helicopter, and we put GPS collars on them to track them in space and time. And so one of the challenges was actually getting properties to work on and capture deer and put GPS collars on them. So given this area is largely private owned, we had to contact many folks to try and get private landowners to give us permission to access their properties. Thankfully, we've had great success and lots of support from folks down there who've been really helpful in getting this project, keeping it going, getting it off the ground and keeping it going. And since for 2018, 2019, and 2020, we've had 3 seasons of capture and release of animals. We've consistently had over 100,000 acres of lands to work on and potentially access deer. Next slide. So this is just an overview. So between 2018 and 2020, we've had 3 years of this study. We were able to put 225 deer on the landscape. We were able to apply GPS collars to them. Most of these animals, for the first year, we did put about 25 collars on adult males, but besides those first 25 in the first year, all animals collared had been fawns, so that we could capture the time when they actually disperse and get an idea of movement propensity of these animals in the landscape. And so as of 10/26, just yesterday, October 26, we've got 60 deer that remain on the air that we're tracking right now. Only 19 from our 2019 cohort and 42 from our 2020 cohort. Next slide. So the deer location data themselves, we have over 400,000 location records. It's an enormous data set, and it continues to get larger and larger because our collars are collecting data every single day on all animals that are still active in the field. So managing all this data is an interesting challenge in itself. And one thing that I want to point out, though, as a caveat, is that the movement information that we do glean from these animals can be incomplete in some cases because the deer might die along their dispersal or migratory path, or the collar might fail, unfortunately. In our first year, we had significant failures of our collar batch, and so we did not get data on a lot of animals that first year. The location accuracy varies for these GPS collars, but we can calculate rates of movement, and we use those to omit spurious locations. So obviously, deer can't travel 50 miles per hour for 2 hours, and so we can easily exclude those kind of locations. And there are also different metrics that the GPS locations provide us to help us vet those locations and basically get rid of bad data. Controlled trials that we've conducted have shown that locations are accurate within 20 meters. So we can, the locations that we do use for our analyses are fairly precise. Next slide. So just some background here, a little bit of information about home range estimates that we've gotten. This is nothing surprising and nothing new compared to the literature about white-tailed deer and what we know about them. The home range that we estimated was about what we expected. For does, the average was about 0.68 square miles. And for bucks, it was 0.85 square miles. And there was a little bit of variance on that estimate, but for the most part, animals stayed within a home range of about 1 square mile, which is no surprise. And they spent most of their time within the forested habitats of southeastern Minnesota. And as folks here on the call probably know, that habitat down there is a mixture of forested habitat and ag. And it becomes more agricultural as you move west. So it's an interesting sort of transition zone that we were working in. The home range increased as a proportion of agriculture within home ranges increased, and this is not surprising either. You know, with less forested habitat, the deer have to have a larger home range in order to find adequate cover. Next slide. This is just an overview of some of the interesting estimates that we derive about deer dispersal and migratory trends. In the figure there on your left-hand side, it shows the southeastern CWD management zone and the control zone and hatch marks there. And the triangles indicate the starting locations for particular animals, and the little octagons, stop sign looking figures indicate the stopping points for dispersing and migratory animals. The only animals that moved over 10 kilometers are displayed on this figure in order to keep it sort of less messy. The red diamonds indicate CWD positive animals. The orange diamonds indicate CWD positive animals in Iowa. And as you can see, we had some movements between Minnesota and Iowa, and we had a couple long range dispersal movements of deer into into the interior of Minnesota to the northwest. Now, the percent of male dispersers was interesting. We estimated 43% of them dispersed in their first year of life, or as yearlings, and 26% of females actually dispersed. And there was a little bit of variance on that because their sample sizes were small to medium. We did see seasonal migrations in very small proportion of males, 6%, and a little bit larger proportion in females, and that really surprised us. And I'll show you some more information about that in a second. But the average movement distances we were observing were about 20 kilometers for dispersal, so roughly 13, 14 miles or so. The median travel date, so when they would actually start their movements, was around May 29, June 1st, and fairly similar for migratory movements. So that's not really surprising based on what we know in the literature. The mean number of days they traveled did differ quite a bit between sexes. So males spent a longer amount of time on their movements, during their movements, than compared to females. But in general, it was interesting because the directionality of their movements tended to go towards the west. Sort of, if you see the figure there on the left-hand side, there is a polar plot there that shows a histogram distribution of movements in 360 degrees. And for the most part, they did move to their west and north. So that was very interesting to us, and to some extent has informed our management and priority of resources moving forward. Next slide. This is just an example of one of the phenomenal animals that we had. This was our longest distance mover, a doe captured in 2018. And we're just going to show you a video real quick. Oops.
Sorry about that, Chris.
It's no problem. Show a video real quick of exactly what her movement looked like. She was able to move from Forestville Mystery Cave State Park all the way up to Cannon Falls through the month of June 2018, and it's about 75 miles, give or take. So can you click? Can you click on the map itself? There you go. Click on that.
Yeah, I just clicked on it and nothing happens.
Oh, it's not.
I'm not sure.
Oh, shoot. Well,
Eric, you might try just clicking on the slide bar and just dragging across. Will it animate like that? No.
No, it doesn't look like nothing happens.
It's got a drag bar, right?
Yeah.
Okay.
Yeah, I'm not sure.
Oh, well, if folks are interested in seeing this video, it just kind of outlines the movement of this particular animal. This video is up on our website, so you can view it there at your leisure. So we'll just skip on the next slide. So that was an example of a long distance disperser, the longest distance disperser we had, a female, which was phenomenal, moving approximately 75 miles. Now, these movements are a little bit different, and I'll take you through these. This is one example of a buck that we captured in March 2018, and it began what we thought was an apparent dispersal just after it was captured in late March. It took about 10 days to move about 22 linear miles, and it went from just north of Granger. You can click, Eric. Go ahead and click. There you go. And there's that yellow circle there. It's where it started out. Click again. And this was the trajectory of movement this animal made between March 21st and 31st, 2018. And then that little red dash circle there is where it ended up just south of Saratoga, Iowa. Next slide. Now, we thought that that was a dispersal at that point in time when we were done, but as we continue to monitor this animal, it trekked back to Minnesota about September 9th, 2018. And you can see the yellow dash line is where it started south of Saratoga. You can click, Eric, and the trajectory of movement almost mirroring what the path that it took coming down. And it remained, took 4 days to get to Minnesota, back to its natal range around where we captured it, and it remained there as of March 24th, 2019. And the problem is it went off the air, unfortunately. So we could not track that animal anymore, and we don't know any further history. So it appears that it could be, and likely was, a migration, but the next example will be even clearer. Next slide. This is another yearling male captured in March 18th, 2018. And if you click the spacebar, Eric, there you go. So it was actually captured around the Granger area in Minnesota, right on the Iowa border. And it apparently dispersed from June 2nd to June 15th. It took about 2 weeks, about 17 miles. And click again, Eric, again to the Saratoga, Iowa area. Next slide. And now, interestingly, surprisingly, it began its first trek back to Minnesota around August 31st, 2018. It took 8 days to cover that ground. It was approximately the same path, again, mirroring its path coming down. And it remained in Granger, Minnesota until June 1st, 2019. Next slide. Then it began its second trek to Iowa on June 1st, 2019. And then took it up 2 or 3 days, a much shorter journey time this time to reach its what we call the summer range, or what we assume is the animal's summer range just south of Saratoga. And it stayed until August 29th. Next slide. And then finally it began its second trek back to Minnesota on August 29th, 2019. It took about 3 days, short time again. Stayed there in the Granger area until October 27th, when it finally went off the air. And this seems to be fairly clear evidence to us of a seasonal migration that was consistent with its timing of its movements between its summer and winter range, which we found very interesting. Now, so there's actually several animals that had similar movements like this between Minnesota and Iowa, which totally blew us away. It's just so interesting. Next slide. And so moving along now, the question arises, okay, well, did any of your collared animals actually have CWD? Well, one of the constraints, we did not test animals when we first captured them. We didn't have the capabilities of doing that, but animals that we did recover from mortalities, we have been able to collect tissue samples and test. It turns out that one of the animals, one of 88 that we've been able to retrieve sample information on, was positive. And this particular animal was a doe that was collared in March of 2018 near Preston, which is inside of our core area of CWD. And unfortunately, the collar went off the air around July 4th, 2018. So we had a very short window to kind of look at the location information of that animal. The interesting thing is that 2 years later, after it went off the air, 2.5 years, October 8th, 2020 comes around and we find this animal dead from an apparent vehicle collision. If you look on the map there, there's a green triangle. There's a green triangle on Route 52 there, just east of Preston, Minnesota, and that's the mortality location. And the green circle there is where we captured the animal. The blue diamonds are all of its locations that we had between March and July of 2018. All the red dots represent the CWD positive animals that we have identified in this area. So it is a hot zone. And the good news, if it's a silver lining at all about this animal, is that it appeared she remained in this area and did not disperse. At least there's no evidence of a migration that we could tell from that data we did have. So hopefully she maintained a very tight home range in here. Next slide. So how do we interpret some of these results so far? Well, it's interesting because we've documented high spring dispersal rates, generally high dispersal rates, and surprisingly migratory behaviors that are extremely important to consider with CWD transmission, especially with our neighbor Iowa, who we know has CWD. Now, dispersal movements can be long, as we have demonstrated with the 75-mile long movement dispersal of one female. Our longest distance buck disperser was about 60 miles. So they can move very far along the landscape. But about 80% of doe dispersers traveled less than 23 kilometers or 14 miles, and 80% of bucks traveled less than 31 kilometers or 19 miles. So that's you know, a silver lining in a sense, because most animals are not dispersing very far. There was a general westward trend of dispersal movements, and that certainly is a concern for westward expansion of CWD. Next slide. And so future directions, we're going to continue to collect data on collared deer that we have in the field until they die or their collars fall off or they stop transmitting. And in all cases, we do attempt to recover the collars to get more valuable information on board the collar. We are expanding the study area to the north and to the west, and we are going to have another capture release session here in January or February 2021, and release another 45 to 50 collars on fawns. And we're going to continue to update our movement estimates and trends with new data that we're constantly being informed by the animals on the air. Next slide. And with that, we can maybe get into some question sessions and some public announcements, announcement slides. Eric, if you want to go through those final ones.
Yeah, so here's both Chris and myself, our email addresses for anyone.
And the official disclosure statement.
Yeah.
Thanks, guys.
Yeah.
We do have a number of questions. Again, I'll try to group them as they make sense and maybe not jump back and forth between, unless you want me to jump back and forth between the movement study and CWD in general. I'll just throw the question out there and you guys can answer whoever you would like to answer. So the first couple questions are around animal disposal. Questions about lined landfills. Where are those located in the state? And does anybody offer high heat incineration? You talked about the 3 different types of disposal that for prions, the high heat incineration, the alkaline digester, and the lined landfills. Do you utilize all 3 of those? And then where might the landfills in the incineration be in the state?
Yeah, certainly. Great question. And so the regulatory agency for landfills in Minnesota is Minnesota Pollution Control Agency. And so their website should have a map identifying the different types of landfills and where they're located. Our DNR dumpsters that we are using They go to either the line landfill or incineration. For the southeast, all of our dumpsters in the southeast are going to the incinerator in La Crosse. And then our dumpsters in the north central are all going to line landfill at the Crow Wing County facility. And the South Metro dumpsters are all going to the, it's a line landfill in, it's called Republic, I believe it's called, but that's a line landfill also. And then when it comes to, when we get a positive deer from a hunter, we contact that hunter immediately with a phone call, and we ask them, and it's to their discretion. If they want to consume that animal, that's up to them. If they do not want to, we offer the option that we can meet them and get all the venison and the if there's carcass remains that they have, say it's still hanging on their meat pole, we'll grab that from them and then bring those items to the veterinary diagnostic lab for alkaline digestion. Agency culling, again, we hold all those animals till those test results come back, and same setup. Those known positives go to alkaline digestion. So we use all three methods, just different context there.
Great, thanks. I think you gave a number of over 90,000 samples across the state since 2002, is that correct?
Since 2002, correct.
Okay, so the question is about getting an idea of movement of CWD into new areas. Is there planned testing outside of the zones that you talked about to see where CWD else might be otherwise? And what about suburban areas, the deer populations in suburban areas?
Certainly, good question. So from 2002 to 2005, DNR conducted surveillance on a statistically statewide based design, and it generated thousands and thousands of samples. But again, it was a shotgun approach per se, sampling across the landscape that way. And so from 2006 and now, we dialed it towards a risk-based surveillance effort. So 3 criteria fit risk-based efforts for us. 1, positive deer farms or cervid farms. 2, any suspect or clinically suspect animal that is reported by the public, we respond to that statewide and get that animal tested. And 3, our adjacent borders. So if Wisconsin has a positive that's close to our border, we conduct surveillance on our side of the border where that positive deer was detected by our neighboring states. And because resources have to be utilized, that's the best approach, is to look at where we know we have risk and to then see how it is in our wild herd. We do, in the back page of the regulations handbook, the 2020 regulations book, the back page, the very last question, it's question and answers. The very last question, it allows hunters that are outside of DNR's surveillance areas, gives them the option to contact the University of Minnesota Veterinary Diagnostic Lab to get their deer tested. There is a fee associated with it that the private public person would have to cover themselves, but that way there is an option that a hunter can get their deer tested outside of DNR's surveillance areas.
Let's jump to a question about the movement study and some thoughts on that. Given the long distance dispersal from Minnesota to Iowa, do you suspect the inverse is happening also from Iowa to Minnesota? And how does the Mississippi River play into that when we're talking about Wisconsin?
Yeah, that's a great question. It's a great question. I wouldn't be surprised. So far we've been seeing those migratory movements between Minnesota and Iowa. It is certainly possible that deer who originate in Iowa may disperse to Minnesota. Absolutely possible. It's too bad we can't get some more samples down there to find out for sure. But you know, the movements that we have demonstrated so far just between Minnesota and Iowa generate, you know, enough interesting questions about risk of CWD transmission between our states that that is definitely a concern. The Mississippi River, obviously is a major barrier, but we know that deer cross the river all the time, especially in winter. We haven't seen any movements of deer between our state, Minnesota, and Wisconsin yet. We're hoping that we're going to try and sample this coming year, 2021, get some samples in Houston County, closer to the border, to see if we can detect any of those types of movements. So that'll be really interesting to find out.
Speaking of migration, why do some deer migrate and some do not? And particularly the deer you're talking about, what's so special about Granger and Saratoga that they're going back and forth?
Another great question. So there's a couple different hypotheses about what could be going on there. One of them that we've been thinking about is obviously the food resources in Iowa during the summer are phenomenal. There's tons of crops to choose from, and it's basically open and free to roam. So they might be spending their summers down there to take advantage of those food resources, and then choosing to come back up to Minnesota where there's perhaps more cover and stay the winter. And there might be some sort of evolutionary element to those movements. So it might be actually ingrained into that particular subpopulation why they're actually moving between those two areas. Could be. But we're looking into some different hypotheses about how density might be related, density of deer might be related to why these deer are moving like that. It's been found in other studies, especially out in Pennsylvania, where a lot of work has been done, that underlying density of deer will influence their decision to disperse on the landscape. So that's a really interesting question for us. One of the caveats to answering that for Minnesota is that the scale at which we have density information is very broad relative to the scale of the deer movements that we see. So we might not be able to fully answer the question. We might not have the sample size to answer that question, but it's certainly something we're interested in looking into.
Good work. You even answered a question I didn't ask yet about deer density and movement. So you knew what the questions were going forward, I guess. Back to some management questions. How do management strategies align with those of other CWD positive states? So how does Minnesota align with other states? And what about natural predators and management of CWD with that?
Yeah, good question. And a lot of the other states are doing a lot of the similar things Minnesota is doing. For example, excuse me, the state of Missouri is utilizing tools as agency-directed culling around known positives to keep two things going: to keep deer densities low in those immediate areas, and to remove additional positive deer where we know there is infection within that section of land. Illinois is also doing similar things with using surveillance to kind of show hunter surveillance to show where these positive deer are located, and then using tools such as agency culling to focus on keeping that disease prevalence low. And Illinois has been doing that for quite some time, and with some success. However, they are seeing some spread as well. Their friends to the north, the state of Wisconsin, their endemic area is just across the border. So, Wisconsin was doing a number of those items, but have taken their foot per se off the gas for a period of time, and and so it's a challenge. It's to keep aggressive tactics. It takes a toll on resources, and it's a challenge. So, but yeah, other states are doing similar things that Minnesota is, and Minnesota talks with other states. We actually have a conference call every third Tuesday of the month with all the Midwest states just about CWD management. So the states are talking and learning from each other what has worked in other states and what are other states doing, and what what Minnesota might consider trying because of other reasons that other states have had successes. And similar stuff with the deer movement study. There are other states doing the similar deer movement studies, and so there's regional collaboration on these studies. So there is big picture, regionally based, and then it dials down to specific picture when it gets into just each state.
So do you have any thoughts, or is there data out there that looks at the management of CWD and natural predators such as wolves?
Well, we know that CWD can make these animals a lot more susceptible to predation, such as from coyotes or wolves. In southeast Minnesota, we don't have, that's not our wolf range, but we do have a number of other predators down there. CWD also makes animals a lot more susceptible to other predators such as vehicles. So we see that as well. I don't know if Chris, if you want to add anything with the predator angle of CWD.
Yeah, I'll add that there's been a lot of interest in that, and some folks out west have evaluated mountain lions and their ability to, or if there is an ability or predisposition to remove CWD positive animals out west. And that if it's folks are interested, they can do a Google search and find that particular scientific article. It's really interesting, or contact me and I'll forward it to you. There is interest too, I know in Midwest, and not in our research group in Minnesota, but not to pick on them, but there are folks in Wisconsin who have talked, research folks in Wisconsin who've talked about the notion that, are wolves actually able to pick out CWD positive animals and control CWD, at least to some level. This is all pie in the sky, and it's just kind of open sort of research thoughts at this point, so nothing formal. But yeah, folks are thinking about that. Realistically, how possible is that predators can, control CWD infection in deer? I think it's probably very unlikely. There's just not enough predators to really handle the amount of deer in general. That would just be my first thought going into it, but certainly something worthy of thinking about and designing a study perhaps to investigate further.
We have a real life application question for the public. If I see a deer on private property that has symptoms of CWD, or it looks like it possibly could be positive, what should I do? Do I harvest it? Do I call the DNR, contact the CO?
Yeah, no, that's a valid question, and a lot of people consider that, especially with the upcoming firearms season. There's gonna be a lot of hunters in the woods. If you harvest an animal, you have to have the appropriate license to do so, and you have to follow the rules of official tagging, so site tagging and etc. So if you come across that situation, my suggestion is, if it truly is a clinical animal, you need to contact the CO, the conservation officer, and they're the ones that will talk to you, and they'll make the decision. And if there's a ticket to ever be written, it's not by research scientists like us with research, but the enforcement officers. So that's the people that you want to call, is enforcement. And even if you call the tip line, they monitor that and they'll get you in contact. If you don't have the conservation officer's number, they'll get you in contact. There is the conservation officer, the finder map on the DNR website, that if you click on the map, it's interactive. If you click on the map, it'll let you know what conservation officer is for that area.
A question back to the movement studies, kind of about management. So any of these results from the movement studies, do they affect any of the management strategies at that point? And how does migration play into CWD management?
Yeah, that's a good question. We're still trying to, I think the first part of the answer is to to say that we need to really better fully understand the migratory capacity of deer and in the subpopulations that we have that appear to be migratory or have some migratory propensity. So understand that process more before kind of, we fully kind of make an evaluation on management recommendations. I can say that migratory animals are very, very probably a higher risk than a dispersing animal, because as long as they're surviving and they're migrating back and forth between winter and summer ranges every year, they can potentially be, interacting with CWD infected animals, become infected themselves, and be transporters, movement back and forth. So it is a very, very, major concern. As far as management, though, there's no way to identify visually, whether an animal is going to be migratory or disperser. So that is sort of problematic. It's definitely something that we'll have to look more into and think about.
A question about movement of animals and carcass movement. Is there data that shows transport of cervid carcasses into states or a meat processor or to a taxidermist? Does that can the prevalence of CWD be traced to those practices?
Can it be traced to the practice? No, I mean, in our situation, when we have a hunter that has a deer test positive, we talk with the hunter and ask what did they do with the carcass. And if it went to a meat processor, then we contact that meat processor to let them know. And a question then is asked to the meat processor: do they batch their deer when hunters bring animals in there? Or do they process individual animals? And so again, DNR does not regulate the meat processors. That's a Minnesota Department of Agriculture. So we work with Department of Ag and inform them of that stuff. So getting back to like the tracing, we don't trace to meat processors. Again, we stress that we know the disease can spread if an infected carcass per se melts into the landscape. That's one way that these prions are deposited at that site. So the more we can mitigate that, take the carcass and properly dispose of it, it's a way to combat this disease. And hunters, it's not DNR doing that. That's on the individual level of the hunter. And so hopefully hunters will understand that a little further and take some onus on that and properly dispose of their deer carcass.
I'm going to leave this with one last question, and it's a philosophical question, maybe philosophical. Based on what the DNR has learned, the Minnesota DNR has learned with CWD in the state, what might we do different if we did not have CWD in our state right now?
What might we do differently if we didn't have CWD?
Correct. So based on what you learned, if we didn't have CWD in the state right now, what might the Minnesota DNR do differently? I'm guessing to keep CWD out of the state.
Gotcha. Well, Eric, you can jump in too, but I mean, my first thoughts are, that going through this experience, I think we've learned that obviously, carcass movement restrictions are a huge potential risk of CWD transmission. Had we not had CWD yet, I would hope that we would, have instituted those kind of restrictions as a protection, to prevent CWD infection. I think that's a big one. And definitely, having a sensitive surveillance system to detect CWD. I think had we not detected it, we still, would be doing such a thing, probably in some sort of in some sort of moving window rotation in different parts of the state that we determine are higher risk, and probably prioritizing those different parts of the state to do that kind of rotational surveillance.
Yeah, I think you're spot on, Chris. You know, the keeping that surveillance active, you know, that those are the ways that the beacons of positive animals shine. And so, you know, DNR can't do it. Again, I go right back. The hunters, landowners, wildlife watchers, help from everybody, you know, and, you know, reporting sick animals. You know, even though a wildlife watcher might not be an avid hunter, they have eyes on the wildlife a lot, and they take photos, and, you know, just being very active in that regards.
Exactly. We're totally, I think that's a great point, Eric. We're partners, you know, with the public here, and, you know, DNR, you know, we're relatively few compared to the people out there. So, you know, it's the hunters and the citizens out there who are paying attention to deer in their landscapes, which are really huge contributors to our success or failure. So another thing I'll mention too is taxidermists, you know, having a very robust taxidermist network to draw samples from, because we know that older bucks tend to have a higher probability of CWD. So in the surveillance context, you know, having an established taxidermist network where we can more easily get those samples to test for CWD and detect it potentially earlier would be a smart thing.
Awesome. Great. Thank you so much, Chris and Eric, for your insight from the Minnesota DNR tonight. We really appreciate it. It's a great way to end our webinar series, being, you know, focused on Minnesota and what's happening here. So thanks so much for being here tonight. Reminder to everybody that this session was recorded and will be available on the MinPro website.